All right, welcome to today's explainer. We are putting a seriously deadly microscopic pathogen right under the lens. I'm thrilled you're here because today's case study, it's just this incredible mix of medical history, brilliant biological deception, and, well, high-stakes clinical investigation. We are decoding a parasitic assassin known as Leishmania donovani. Now, to really understand why this pathogen demands our absolute respect, you just have to look at the clinical gravity of the disease it causes. We're talking about visceral leishmaniasis, or as it's historically known, Kala-Azar, or black fever. It's a severe systemic disease. And the stakes here? If left untreated, we are literally looking at a staggering 75 to 95% mortality rate. So yeah, we aren't just talking about a mild infection here. This is a microscopic killer that systematically dismantles the human body's defenses. Okay, let's dive into this. Here is our roadmap for today. First, we'll cover discovery and taxonomy. Then, morphology, the two forms. Third, we'll track the parasite's life cycle. Fourth, Kala-Azar, the black fever. And finally, diagnosing the infection. All right, Chapter 1, Discovery and Taxonomy. The story of actually identifying this pathogen plays out kind of like a historical medical mystery, taking place across completely different corners of the globe. So picture this. The year is 1900. William Boog Leishman is examining a spleen smear from a soldier who tragically died of Kala-Azar at Dum Dum, near Kolkata. Leishman spots this totally unique parasite. Then, just 3 years later, in 1903, Charles Donovan is independently examining a patient over in Chennai, and guess what? He reports finding the exact same parasite in a spleen smear. Because of these parallel independent discoveries of patients dying from the exact same mysterious fever, the scientific community ultimately mashed their names together to identify the culprit, Leishmania donovani. So what exactly is this suspect we're looking at? Well, checking out the pathogen's profile, it belongs to the family Trypanosomatidae. It's an obligate intracellular protozoan parasite, which basically means it absolutely must hijack and live inside the cells of its host just to survive. Specifically, it targets the reticuloendothelial system. Think of that as essentially the body's network of immune sentinels. It literally hunts down the macrophages and monocytes in our liver, spleen, bone marrow, and lymph nodes. And its primary getaway driver? In the old world, so Asia, Africa, Europe, and the Middle East, it relies entirely on a vector to get around, the female phlebotomous sandfly. Moving right along to Chapter 2, Morphology. The two forms. Now, what's really interesting about this is that to survive the jump from a flying insect into a human being, this parasite has evolved a masterful biological disguise. It doesn't just have one shape, it has two. On the left, we've got the amastygote form. This is the intracellular stage you find inside human hosts. It's tiny, just 2 to 4 micrometers, round or oval, and completely non-motile. But then on the right, we have the promastogote form. This is the extracellular stage found in the digestive tract of the sandfly. It's elongated, spindle-shaped, much larger, like 15 to 25 micrometers, and it's highly motile, thanks to a free anterior flagellum just extending right out from its body. It literally shape-shifts depending on which host it's living inside. How crazy is that? So when we're trying to actually spot the human-infecting amastygote form under a microscope, we use a specific chemical stain, like the Giamsa stain. When we do that, the delicate cytoplasm of the parasite turns this really striking purple-blue color. The central nucleus stains a distinct red. But the real giveaway? That would be the kinetoplast. It's an organelle sitting right next to the nucleus, and it shines a brilliant, deep red. That bright red dot right there, that is our microscopic smoking gun. Okay, Chapter 3, the parasite's life cycle. Let's move to and see how this actually builds, step by step. It all starts when a female sandfly takes a blood meal from an infected human, ingesting those tiny amastygotes. Inside the sandfly's midgut, those amastygotes transform into the long, motile promastygotes and just rapidly multiply. These promastygotes then migrate forward and, get this, they actually block the sandfly's feeding canal. So when that blocked, probably very hungry sandfly bites its next human victim, it's basically forced to regurgitate these promastygotes directly into the skin wound. The human body naturally responds by sending immune cells called macrophages to gobble up the invaders. But once inside the macrophage, the promastygote simply transforms right back into an amastygote, multiplies by binary fission, and eventually bursts the cell wide open to infect even more cells. And the whole cycle repeats. I really want to pause and emphasize the sheer biological cleverness happening here. It's literally a microscopic Trojan horse. Human macrophages are designed to eat and destroy pathogens, right? But Leishmania donovani actively wants to be eaten.
It survives the super hostile environment inside the macrophage, effectively turning our own immune system's primary defenders into its own microscopic replication factories. It's absolutely brilliant and terrifying.
Which brings us to Chapter 4, Kala Azar, the Black Fever. Pivoting from the microscopic to the macroscopic impact on the human body, the crucial point here is understanding how those bursting macrophages actually translate into systemic failure. The incubation period can vary wildly, anywhere from 10 days to 2 whole years. Once it's active, patients suffer from prolonged high fever, severe weakness, and rapid weight loss. And because the parasite specifically targets the reticuloendothelial system, we see massive splenomegaly, a hugely enlarged spleen, and hepatomegaly, an enlarged liver. Also, the skin on the face, hands, feet, and abdomen often darkens to a sort of grayish hue, which is exactly what gives the disease its namesake, Kala Azar, or black fever. Furthermore, the parasite invades the bone marrow itself, causing severe suppression. This leads to profound anemia, leaving the patient pale, weak, and pretty much stripped of white blood cells. And that bone marrow suppression, that's exactly why we see this terrifying 75 to 95% mortality rate in untreated cases within 2 years. But here's the catch. The most common cause of death actually isn't the parasite itself. It's secondary bacterial infections. The immune system becomes so severely weakened, so depleted of white blood cells, that the body simply cannot defend against just everyday run-of-the-mill bacteria.
Now, in modern times, this presents an incredibly tragic complication. Visceral leishmaniasis has become a major opportunistic infection for people living with HIV/AIDS. There is this absolutely deadly synergy between the two. HIV increases the patient's susceptibility to Leishmania, and in return, the visceral leishmaniasis dramatically accelerates the immune suppression caused by HIV. They basically assist each other in totally destroying the host's defenses.
Let's move on to our final chapter, Chapter 5, Diagnosing the Infection. So, how does modern science actually track down and confirm the presence of this microscopic assassin in a patient? Well, lab scientists have a few main options, and this brilliantly illustrates how far we've come. Microscopy is fast. Using Giemsa stains, we can visually confirm those amastigotes in tissue samples. However, morphologically, they look pretty much identical to another parasite, Trypanosoma cruzi. The more conventional diagnostic approach is isoenzyme analysis through culturing the parasite on specific media. But the problem there, it takes several weeks. Time a severely ill patient just might not have. And this is exactly why PCR, or molecular diagnosis, is the superior modern choice. Within days, PCR can amplify a specific genetic segment, the rRNA internal transcribed spacer 2, providing highly sensitive, rapid identification down to the exact species of Leishmania. Just a quick professor's tip for anyone out there studying infectious diseases. Avoid relying heavily on serology or antibody detection for this one. It honestly has very limited value because most patients with cutaneous forms just don't develop a significant antibody response. But more importantly, the antibodies can cross-react with Trypanosoma cruzi, meaning if you're treating a patient who has traveled through Central or South America, an antibody test could easily trigger a false positive, totally sending your diagnosis down the wrong path. So definitely watch out for that. So we've tracked Leishmania donovani from its historical discovery in military outposts all the way to its molecular DNA signature in modern labs. We've seen its brilliant 2-stage disguise and its absolutely devastating toll on the human immune system. But this leaves us with a pretty critical question for the future. As global travel increases, and as changing climates potentially shift the geographic range of the phlebotomist sandfly into entirely new regions, where will this microscopic assassin strike next? Think about it. Thank you so much for joining me for this explainer. Keep questioning, keep learning, and I'll catch you next time.